In the last video, I talked about a problem for the discovery process, which is that individuals tend to be biased. That is, even smart, well-meaning individuals can hardly help but bring some biases to bear on the questions that they're asking about the world. So in this video, I want to talk about the credibility cycle, which is the part of the scientific method where we try to deal with the individual biases. So if you remember the two conversations model with the two circles in it, the picture that's here is Grinnell's sort of expansion of the credibility process. So you can see once the individual researcher or research group produces a journal article, they send it out to editors and reviewers, and we'll talk about that. That once it gets published, it goes out to a broader research community, then other scientists, maybe into textbooks, and the broader public. So this is all a long process of increasing degrees of, hopefully, intersubjective agreement. So when this process goes right, the further something gets through the credibility process, the more intersubjectively verified it is, and therefore less biased by individual perspectives. Okay, so the credibility cycle process starts with peer review. I hope you're familiar with this. Peer review is the process by which other scientists evaluate the submitted journal article. So you write up your discovery claim, your proto-science, as Grinnell puts it. You send it to a journal, and then the journal puts it through the peer review process. So at first, editors read it. Typically, they kind of glance over it to make sure that it looks kind of like a scientific research report. Scientific journals get some wacky stuff sometimes, so it's important that the editors should do a kind of pre-screening process. And if it passes that stage, it usually goes on to 2 or 3 external reviewers. So people who are hopefully in the field of the paper being evaluated, they know the paradigm that the person's working in, and they can see whether the journal article that's been submitted meets the kind of disciplinary standards established in that field. They will either accept the paper, request revisions to it, or maybe request some new experiments be done, or reject it outright. Okay, so the peer review process is deeply paradigm bound. So the peer reviewers, the people who are reviewing the article, typically don't have access to things like the raw data, and they don't typically have access to the notebooks that the scientists were working with. So if you remember, Grinnell describes a journal article as a cleaned up version of the actual process of scientific discovery. It's the story that you would have told if you knew what you were looking all along. So that cleaned up story is what the peer reviewers receive. They don't typically, sometimes, but typically they don't receive the raw data they're working with or the code that they use to process that raw data. All they see is the sort of finished product of the discovery claim. Now, what that means is that they can only ask themselves, does this look to me like it fits the methods and experimental standards that are typical in my field, right? So they're not asking themselves, they don't have access to whether the experiment was properly run, like did they use the machines right, did they record things properly. All of that is in the notebooks of the scientists, but it's not in the journal article. So really all they can do is ask themselves, does this match the paradigm that we have for what counts as a good experiment in our field? Okay, so of course, the idea here is that some of the biases of the individual researchers won't be shared by the peer reviewers. And with any luck, the peer reviewers will sort of make sure that the individuals involved aren't being biased in any important way. It's common to think that groups of people are actually less rational than individuals, but there's a counter narrative to that we could consider, which is that sometimes groups are in fact more rational. So here's just a nice example from the early 1900s. So Francis Galton, in a paper published in the journal Nature, recounts this anecdote. So in 1906, Galton goes to a county fair and gets the results of a contest. And the contest was guess the weight of this ox. So here's a picture of an ox. So about 800 people submitted their guesses, and whoever got the closest won a little prize. So Galton collected up all these guesses and did a kind of statistical analysis on them. And what he found was that the median guess was about, so if you take all the guesses and add them up and then divide by the number of guesses, the median was about 1,207 pounds. The actual weight of the ox was about 1,198 pounds. So that's within 1% of the real value. Now, individual guesses varied widely, and very few people got within 1% of the actual vote or actual weight. So it seems like in this case, by aggregating the opinions of a bunch of people, you actually get a more accurate picture of reality than just asking any individual person. So broadly speaking, that's the idea of peer review, that the more eyeballs you have looking at a discovery claim, the more likely it is that you'll be able to cancel out individual biases. Now, as I said, the typical peer review process doesn't involve 800 reviewers. It involves 2 or 3, normally. Sometimes more people than that get involved, but the normal peer review process is much more limited than that. So, but nonetheless, the process continues. Even after it's past peer review, even after a journal article is published, the credibility process doesn't stop. Now, as I said, the peer review process mostly happens via checking whether the paradigm for what counts as a good experiment is met by the purported discovery claim. Now, paradigms have to kind of constantly shift. They have to constantly be updated. So some standard of experimental evidence might not be sufficient. It might turn out that you need a higher standard or a different standard or something like that. So here's one of my favorite examples of this. This is an image from a paper titled Neural Correlates of Interspecies Perspective Taking in the Postmortem Atlantic Salmon, an Argument for Proper Multiple Comparisons Correction. Okay, so neural correlates of interspecies perspective taking in the postmortem Atlantic salmon. What these researchers did was the following. They were trying to calibrate their machine. So they've got these MRI machines that are measure brain activity. So they measure the amount of blood flowing in different parts of the brain, and they wanted to just calibrate their machine. They weren't actually doing an experiment. So they took a dead salmon, completely dead, and put it in the MRI machine, and then did some of the things that they would do in a normal experiment. Like they'd show it a picture of somebody else. Well, they put a picture in front of the dead salmon of somebody else, like looking at something. So interspecies perspective taking. And what should have happened is that there was no MRI, like there was no brain activity detected in the salmon, right? It's dead. It's not having any brain activity at all. But what they found was they got 2 or 3 points at which there was statistically significant brain activity in the dead salmon. So the conclusion here shouldn't be that dead salmon have some response to seeing pictures of other species, right? The response should be there's something wrong with the way that these experiments were being done. So they used all of the standard setups, all of the standard statistical techniques in their field that were part of their little paradigm, and they found a result that they know can't be true. So what that shows them, and what they argue in this paper, is their paradigm needs updating, right? You need to do something better than this if you're getting brain activity in the brain of a dead salmon. Okay, one more example of this, particularly sort of earth-shaking one in the field of psychology, was this paper published in 2011 by Daryl Bem. So just to read you a bit from the abstract. So it says, this article reports 9 experiments involving more than 1,000 participants that test for retroactive influence by time reversing well-established psychological effects so that the individual's responses are obtained before the putatively causal stimulus event occurs. Okay, so what Daryl Bem is doing in this 2011 paper called Feeling the Future, is taking extremely standard psychology experiment paradigms, little sort of like ways of doing a psychology experiment, and just putting the stimulus after the response. So you might have a paradigm like this, a priming effects paradigm, which is like you show somebody a list of 10 things that are sort of farm related, and then they're slightly faster at recalling or producing farm related words. It's a priming effect. Now, what Daryl Bem did was just ask them to recall or produce a farm related word and then show them the list of farm related words, and found that if he afterwards showed them something that was related, that he would get the priming effect. So, I mean, this is just sort of the flavor of what he was doing. He did a bunch of different experiments with lots and lots of participants. They were statistically well powered. They met all of the standards for a psychology experiment at this time. So he absolutely matched the paradigm or sort of like lived up to what the paradigm says the standards of evidence and the type of experiment should be. But what he was finding is people seem to be able to read the future. Okay, so there are two things that you can do with this result. You can say, A, I think that people can feel the future. That's one thing that you could take away from this. Or B, you could say, there's something wrong with our paradigm. These experimental methods are not sufficient to tell truth from falsity. Daryl Bem was one of the people in the first camp. He said, look, I think that I have actually shown that people can sense the future. Almost everybody else said, what you've demonstrated here is that psychology is broken in some deep or important way, that our paradigm needs updating. So one of the things that may have been wrong with this is that he would do this process. So this is a quote from a Slate article. He would do the following. He says, quote, I would start one experiment, and if it just wasn't going anywhere, I'd abandon it and restart it with changes, Bem told me recently. Some of these changes were reported in the article, others weren't. Quote, I didn't keep very close track of which ones I discarded and which ones I hadn't. Okay, so what he's probably doing here is starting an experiment, and if he's getting data that happens to show people feeling the future or apparently feeling the future, he keeps running it. If it just shows nothing, then he stops. And nobody's quite sure how many experiments he ran. These happened over years and years and years while he was quietly having a very successful, he was doing these experiments kind of quietly, not secretly, but he wasn't advertising them very much. But he had a very successful psychology career outside of this. So he's a well-regarded psychology researcher. Everybody agrees that he knows the paradigm inside and out. But he was being a bit selective about which experiments he kept going with and which ones he threw out, which apparently wasn't part of the paradigm. So it was acceptable practice at that time in the rest of psychology to, if your experiment's not working, you just throw it out and move on, rather than rejecting that you found some, disconfirmation of your hypothesis. So there's probably other things that were sort of issues here. It's a big complex debate that's been going on in psychology ever since. So people took this very, very seriously as a call to rethink methodology. And lest you think that this is me doing a knock on psychology, I think this is actually science at its very best. So when scientists are confronted with something that shows them that their paradigm is insufficient, the very best thing they can do is try to revise it, right? Rather than ignoring this and saying, it's a one-off, psychologists have genuinely risen to the challenge and done a bunch of stuff to try to tighten up their experimental methods. Okay, so that's part of this next step. So this little story I just told you about Daryl Bem and Feeling the Future is after something gets published, that's by no means the end of the story when we're thinking about how credible a discovery claim is. So because Daryl Bem followed all the rules of psychology in 2011, he got this published. I promise you, people looked really hard at this paper trying to find some reason to disqualify it, but they couldn't find any. So nonetheless, after it's published, people the broader research community really had a hard think about what it meant. So they had to evaluate the significance of that publication and think hard about what its consequences were. Now, in this stage, when it reaches the research community, a couple of things can happen. This Daryl Bem story is a pretty dramatic one, which is why I like it. Usually, people's experiments don't kind of change the paradigm that they're existing in. Very often, a research article is cited a couple of times and then ignored. One of the best things that can happen to you as a scientist is that your research article after publication gets cited a lot. So what that means for you is that your discovery claim is shaping future science, and that's one of the metrics that they use when they're granting agencies and other people that hand out money to do science. One of the metrics they use is how many times has your journal article been cited by other people, which is a sort of proxy for how much of an impact did this make on science going forward. Okay. So one of the ways that we transform a single discovery claim into a more credible one that we consider to have better intersubjective validity, is by doing a meta-analysis. So typically, one journal article shouldn't be regarded as proof of anything. So, one journal article shows that you got this result in your experiment, but it's not obvious just from that we should conclude that the thing that you're saying is true. So being able to show something over and over and over again, that's supposed to be the hallmark of good science, right? So a meta-analysis, I think you kind of get the picture from this picture, if you can see it. A meta-analysis takes a whole bunch of studies on the same subject and tries to combine their data into one big meta study, right? It looks at, we've got 300 studies on the efficacy of vitamin C for fighting a certain type of disease. We're going to look at what all of them say and do a statistical analysis across a bunch of studies. So this is a way of sort of situating each individual discovery claim in a bigger picture to try to get a sense of, what it all means added up. So this is the gold standard for trying to evaluate the significance of individual studies. It's got some issues. For example, it's not clear what to do with studies that are underpowered, by which I mean, so suppose you're trying to evaluate the significance of vitamin C for the common cold, and your study only had 10 people in it. So do you include that in the meta study or don't you? It's data, but it's data that maybe you did a Daryl Bem on. Maybe you did 15 studies, all with only 10 people on it, and this is the only one that showed the result you're looking for, and you just didn't publish those other ones. So just because this data doesn't mean it's helpful at getting us closer to the truth. So there's real questions about how good does a study need to be included in a meta study. Furthermore, there's really serious questions that go beyond the scope of this course about how do you combine studies with different methodologies. So each of these studies is going to be asking slightly different questions because journals don't really like to publish the exact same study. Like if that exact study has been done before, it has very low novelty. And journals like really novel, interesting claims. So you very rarely get an exact replication of some previous study. That means that every one of the studies in this meta-analysis are asking slightly different questions in slightly different ways. And it's just not clear how to combine all of those. Like if you were doing the same study 50 times, then you can simply add them all up. But that's rarely the case because of the kind of incentives of trying to publish something in a high-profile journal. High-profile journals like novel results. So those are some serious questions about meta-analysis. Here's another one. So there's a serious question about what gets into the literature in the first place. This is called the file drawer effect. The file drawer effect is, again, driven by the kind of incentives that journals have. So a journal wants a novel result, and they really like positive results too. So if you try a big, long study and just don't find anything, it could be because you're bad at science. Seriously, it could be because there's no effect to be found, or it could be because you didn't set up the machine right, or you didn't know what you were doing, or your grad students had a bad week and they just messed it up. So positive results are much more likely to get published than negative results. But what that means is when you're doing a meta-analysis, you're drawing from a whole series of studies that happen to work out. And it's not clear that that's going to be an unbiased sample of reality. So meta-studies are working with essentially a biased set of data because journals really only want to publish positive results, not failures to replicate. And meta-analyses only work with the published literature. They don't work with just everything that everybody's ever done in a lab. So, this is this adorable little cartoon here. It's easy. First you shoot the arrow and then you take your paintbrush and draw the target around it. So, pre-registration is one of the ways that people are starting to think about trying to mitigate this file drawer effect so that our meta-studies work better. So pre-registration is when you register in some publicly accessible database and you say, I'm going to do a study about vitamin C's effect on the common cold. Here's what my methods are. Here's how big of a sample I'm looking for. And when you do this, you encourage the reporting of negative results. You might not get it published in a big fancy journal, but at least it's out there, right? At least you got it out there that you tried to find an effect and didn't. So pre-registration, that's one of the strategies for trying to mitigate the file drawer effect. There are, inspired by the Daryl Bem debacle, we might call it, there has been a movement in psychology particularly, but in a bunch of other disciplines to try to do big replication studies. So attempts to just precisely replicate past results. And that's been helpful to sort of help pick out which studies were genuinely reliable and which ones weren't. No. Okay, so, and then the final step here, after you got it through, suppose you've got a discovery claim, it's made it through peer review, it's made it out to the community finds that it's been credible, they've cited a whole bunch. Maybe if you're really lucky, you get to go into a textbook. And we talked earlier about textbook facts, how they're the kind of shiniest, most finished product of science. And then via textbooks, you get out to the public. So that's how you learn science, typically through textbooks. Okay, so let's talk a little bit about something that jumps the queue a little. So science journalism, depicted here as a dumpster fire. Very often, and I love science journalism. I find it very nice. Every day I read the regular news and it's all full of horrible stuff. And every day I read science news and it's full of interesting stuff that we figured out about the universe. It's very uplifting to read. However, it kind of skips a few steps in this credibility process. So it goes from sometimes just the submitted manuscript and it jumps over the whole community evaluation step, the whole research community, the broader other scientists, getting published in a textbook, and goes straight to the public. So this is good because it keeps the public engaged in ongoing research and presents enthusiasm and is interesting. I love reading science news, but it also has some serious downsides, some serious issues. And I'd like to just walk through one example of this, and I'll note that this is an example that's quite like one of the essay prompts for the first short essay. Okay, so this is from ScienceDaily.com. This is one of the good science journalism outfits. Like this is not a fly-by-night operation. They're good at what they do. I read their stuff all the time, so I quite like them. But you see this kind of thing fairly regularly. So, there's a catchy headline. Beauty sleep could be real. Oh, okay, I'm interested in that. I want to be attractive, so tell me more. And then there's usually a slight difference, or sometimes a substantial difference, between what's in the article and what's in the headline. Headlines and, say, the pictures are chosen typically by an editor, whereas the actual content of the article is written by a science news journalist. Now, science news journalists are often earnest people. Like, you don't get into science journalism because it's a super easy way to make a ton of money. You get into it because you love science and you want to communicate it to a broader public. So the science news journalist has typically done their very best to represent the research in some interesting, accessible way. And then their editor puts a picture of a attractive woman in an expensive looking bedroom and puts a catchy headline like "Beauty sleep might be real." Okay, so the content of the article. Biologists from the University of Manchester have explained for the first time why having a good night's sleep really could prepare us for the rigors of the day ahead. The study in mice? There's a picture of an attractive woman on the headline. They didn't say anything about mice. Okay, the study in mice published in Nature Cell Biology shows how the body clock mechanism boosts our ability to maintain our bodies when we are most active. And because we know the body clock is less precise as we age, the discovery, argues lead author Professor Carl Cadler, may help one day unlock some of the mysteries of aging. Cool. That sounds interesting. So, not exactly that beauty sleep is real. It could prepare us for the rigors of the day ahead. Nothing about beauty in there. It's more about, being active. But okay, cool. So there's a step from the headline to the article. The article is written by a different person than the headline with sort of different motivations. Fine. So now let's take a look at the journal article. Good Science News. You know you're looking at Good Science News because they link you to the article or somewhere where you can read more in detail about what's actually going on. So the article that the journal article that this Science News article is based on is called "Circadian Control of the Secretory Pathway Maintains Collagen Homeostasis." Less catchy than beauty sleep might be real, I admit. Here's what they say in the headline. Here we show circadian clock regulation of endoplasmic reticulum to plasma membrane procollagen transport by the sequential rhythmic expression of SEC61, Tango 1, PDE4D, and VPS33B. The result is nocturnal procollagen synthesis and daytime collagen fibril assembly in mice. Okay, so what they actually did in this study was take some mice who've had their circadian rhythms genetically knocked out, and then studied their collagen. Collagen is a kind of connective tissue, and found that knocking out their circadian rhythms is bad for their collagen. Okay, so I hope you can see we've come a really long way from beauty sleep might be real to this thing. There's they're not, it's not like they're lying, but we've really gone a distance from the actual content of the discovery claim, which I'm going to be honest with you, I don't completely understand what they're saying. I thought I understood what this was about way back when it was a headline. Here at the article level, I'm like, okay, I'm following. When we get to the study, I'm lost. Like, I don't even know what these people are talking about. But I read the thing, and I can tell you it's about knocking out the circadian rhythms of mice and seeing what it does to their collagen. So it's a real journey. So if you go on this once in a while, so open up a Science News article from the catchy headline, compare that to the article, and then compare the article to the actual journal article, it can be a real journey, let's say. Okay, so, what they've done here is essentially skipped this really long pathway from having a published journal article. Usually science journalists are sort of calling the newest publications, looking for interesting stories. and they've skipped the whole credibility process from there on to just reporting it, and they're just reporting it in a way that's as clickbaity as possible. Okay, that's it for this week. Next week we'll talk about scientific integrity.